Today, we will walk through the Bio-Rad rare cell analysis protocol, beginning from software installation through an experiment set up with cell filtering, identification, and creation of your final report. If you do not yet have rare cell analysis, the software and related dependencies can be downloaded from Bio-Rad's website on the Genesis instrument page under Downloads. Within the downloaded folder, first install the certificate file. Select Local Machine and click Next. Select to place all certificates in the following store, browse and select Trusted Root Certification Authorities. Confirm and finish the installation. Once the installation is complete, navigate into the folder labeled Dependencies, then x64. You will need to install two dependencies: Microsoft Net Core Framework Debug 2.2 and Microsoft Net Core Runtime 2.2. Select each and install. Microsoft UI XAML 2.4 and Microsoft VC Libs x64 are optional dependencies if internet connection is not available. Once completed, return to the main folder and install the application bundle file labeled Rare Cell Analysis UWP. Select Launch Settings and toggle on to allow file access. Launch or open the Rare Cell Analysis app and accept the terms of service. Once opened, you can optionally save the app to your taskbar by selecting Pin to Taskbar. To begin a new experiment, launch the Bio-Rad Rare Cell Analysis software. We will walk through this protocol starting from New Results file. Enter your username, the sample ID, and slide ID from your experiment. Then optionally select the source file for your template. A template is a set of imaging parameters that you can customize and save as a file. If you do not yet have a template file, refer to the user guide for more information. Once all fields are complete, select Proceed. You will be taken to the template file configuration page preloaded from your template file parameters. Confirm the experiment details and define the imaging source files for each channel. Beginning with DAPI, select the source folder, navigate to your experiment stitched image folder, and select the associated DAPI image. Continue this process for all channels. Cell identification parameters, such as defining diameter in microns or fluorescence intensity, can be edited to expand or refine identified objects for analysis. Once complete, select Proceed to begin analysis processing. After the analysis has completed, click Proceed on the confirmation screen to continue to the Target Cell Lists and Graphs page. Here you can visualize identified target cells in a graph or list and set channel filters. Selecting the green 3 dots on the left-hand side will open a navigation pane for navigating through experiment pages. We will now walk through this example dataset for circulating tumor cells, or CTC, selection by DAPI and pan-CK positive, CD45 negative cells, and generate a final report. To begin, note the initial count of identified target cells. However, this does not indicate that all identified cells are true CTCs. Navigate to the Target Cell Confirmation page. On this page, identified target cells can be manually filtered by highlighting the row of a cell of interest. Highlight the row by selecting anywhere within the first column other than the checkbox. This will allow navigation up and down through the list using the keyboard. If a signal is identified as debris or an artifact such as the signal seen here, select the checkbox or simply press the spacebar on your keyboard to deselect this cell from the identified target cell list. Note how the total cells have decreased. This list can be optionally navigated by row or through an applied filter, which we will demonstrate. In this example, we want to identify CTCs by a positive DAPI and GFP signal, but negative Cy5 signal, indicating the presence of nuclei and pan-CK, but negative for CD45, a white blood cell marker. To do this, we will navigate through this list until we have identified a representative population expressing the signal we want to identify and mark their ID number down. Once representative cells are identified, return to the Target Cell Lists and Graphs page. Select the identified cells from the Target Cell List tab. Note how these cells are highlighted blue on the scatter plot. Identify how these cells with our target expression cluster according to pan-CK in GFP and CD45 in Cy5. Set the plot y-axis to fluorescence GFP and the x-axis to fluorescence Cy5. This allows us to visualize how our targets and similar targets cluster. Ideally, a break in the cluster data on the Cy5 axis can be visualized for filtering out high Cy5 expressing targets, as the goal is to target cells with low Cy5 expression. In this dataset, the visual break on the Cy5 axis is approximately 6,000. Return to the Chart tab on the left and navigate to the Cy5 channel. Check the Cy5 fluorescence parameter on and define the maximum to 6,000. Select Refresh Filter on the graph. Notice how the new identified target cells only include those within our set filter. This method can be applied to similar filtering goals using the target cell list and chart parameters. Now, with our refined identified target cell list, return to the Target Cell Confirmation page to complete cell confirmation. Highlighting the first row, use navigation to determine if the signal seen represents a true CTC cell versus debris or artifact. A true CTC signal that we are looking for will have DAPI and pan-CK be negative for CD45 and demonstrate expected cell morphology. Here you can see examples of debris and artifacts, which we want to remove from our confirmed cells by deselecting the checkbox of that row or pressing the spacebar on the keyboard. Some wells may exhibit CTC clusters or multiples, as you can see here represented by CTC expression, but presence of multiple DAPI signal, suggesting the presence of multiple cells or a cluster. In this case, as the object is registering as one cell, a comment can be added by selecting the comment box in this cell row. Write your preferred comments such as +1, multiple, 2 CTCs, or similar to clearly record the true number of cells within this row for the final report. At any point during analysis, a results file can be saved for records, to work on later, or to transfer the file to another workstation with rare cell analysis. To save the results file, select File in the top left corner and Save Results File. Select your save folder and file name. To open a previous results file, begin from Launch of Rare Cell Analysis and select Open Previous Results File. Select the file of interest to open the document information page. From here, use the navigation pane to continue working on the file, as selecting Proceed from the document information page will reinitiate data processing and overwrite previously applied filters and cell identification. When cell confirmation and final analysis are completed, a final report can be generated. Navigate to the Target Cell Confirmation page. Select the gear icon in the top right corner to define the report file settings. Set the company or institution name, an optional website, address, or any additional information, and the report title. Page numbers are optional inclusions as well as the report logo. A logo, such as a company logo, can be uploaded by selecting the white box and selecting the logo image. A save location must be defined by selecting the folder icon and selecting a save folder for the report. Once the report settings have been completed, select Apply, then select the Create Report button in the top right corner. The final report includes parameters such as the total number of cells identified, target cells identified, and cells confirmed. Each confirmed cell row will be reported, including any added comments. For multiple CTCs, easily use the search function to find added comments and optionally add to the total cell count. This concludes our training for the Genesis Rare Cell Analysis software. Thank you for joining us.